to the different isogenic seed lines of wheat cultivars. After 7 days of sowing, that conidial spores of BGT spore inoculated by using the sterile lancet needles. And the observation of the disease screening after 7 to 10 days of post-inoculation, we are observing that based on the scale 0 to 9 scale and 0 to 4 scale based on the leaf area covered by the moss of powdery mildew conidial spores. That conidial spores was collected by gently tapping and freshly from the freshly infected seeds into the new and sterile transolution capsule tubes. And BGT conidial spores were further used for DNA extraction for molecular analysis to identification of virulence pathotype and confirmation of virulent pathotypes. So now this resultant discussion part, we are identify the highly virulent isolates from the 150 isolates of 150 isolates of wheat powdery mildew pathogen from our institute, that southern hill zones of Nilgiris and other regions of northwestern plain zones and north hill zones also. Some group of virulent BGT isolates have to be identified and used for rapid screening for wheat genotyping within 15 to 20 days under the specifically designed control conditions at Wellington at the Nilgiris in Tamil Nadu. These are all the techniques we are used. This is all the controlled condition system for polyols for only maintained powdery mildew spores in this. And the second image shows that differential sets for identification of wheat virulence pathotypes for powdery mildew. And these are the 3 different sets we're using for this identification. And this powdery mildew single colony sporulation in wheat susceptible and resistant cultivars. And this is a spore collection by tapping methods. And this is a transolution tube to collect the spores and contain the spores to avoid the external contamination from other bacterial contaminations. And this is all the molecular work further we have to do with identification and confirmation of virulence isolates. Thank you.
Open for discussion.
वो तो जो कमेटी मेंबर है उन लोग करेगा, कमेटी मेंबर कुछ नहीं करना, पूछेंगे अभी इवैल्यूएशन कर रहे हैं ना सब लोग.
Dr. Naladambi?
Yes, Dr. Devanathan.
Yeah,
Yes.
question
Yeah.
answer.
So after this, you can ask the next presenter if they don't have any questions, because
Okay. Give us next. Go to next presentation. You can evaluate on the presentation.
so I request the jury committee members to kindly evaluate each presentations.
Okay.
Even I think Dr. Nandatham, you can ask, huh? The
Jury can ask also questions.
Yeah.
for any clarification. Jury
No,
can
no.
ask.
Otherwise, general people also they can ask 2 minutes for discussion. Na, we kept.
Mostly, sir, the jury will ask.
Generally, for posters, the jury.
Any
No. Any audience can ask, because we kept 2 minutes for the discussion, 5 minutes for presentation.
Okay,
and
okay,
2 minutes
okay,
for
sir,
discussion. Yeah.
you any powder mill listing identify for future breeding program? Arti. Is a successful powder mill question line, promising line you have?
Party has left.
Aarti, you are here?
Aarti को unmute करो.
Aarti?
Hello, sir.
You have any promising powder mill question line?
Yes, sir, we have
Out
it.
of which one is the most convincing one for breeding program?
No, sir.
No question. Did you understand question? Dr. Devanathan, will you repeat your question, please?
Sir?
have identified any promising powder mildew resistant lines for future breeding program, out of which?
No, sir. No, sir. We have to do.
Oh,
I think this is only the protocol.
as the concrete solution.
Yeah.
Okay. Okay. Next one.
Next.
Addition.
Yes. So how many posters will be presented now in this session?
I think another 13 or 14 is there, but we do not know how many people are present here for presentation.
Yeah. That's why
Yeah.
if you can spell out the names, that will be better one by one.
No,
If somebody
Next
is
one, Agisha, sir. Agisha.
We have the list. We already posted
Yes.
the list.
Agisha, are you here?
Yes, ma'am.
Yeah, please.
because the participants may not be knowing, that's why you should announce time to time the name of the participants.
Development of GFP tags.
Shall I start, sir?
Visualize the plan, organization.
Shall I start, sir?
Hello.
Hello, sir.
Okay, start. You can start your presentation.
Okay, sir. Respected sirs and madams, good afternoon and all. I am here to present my research findings on the study development of a GFP tagged Sporosaurus sitaminum strain to visualize implantar colonization in sugarcane. Coming to the introduction, sugarcane smut caused by the Basidiomycetes fungi, Sporosaurus sitaminum is one of the most important diseases of the crop worldwide. The most characteristic symptom of the infected plant is the development of a whip-like sorus composed of plant and pathogen cells modified from the apex of the primary meristem. The life cycle of Sitaminum consists of 3 distinct stages: diploid teleospores, haploid sporidia, which are of 2 mating types, MAT1 and MAT2, and dikaryotic mycelia. The present study is attempted to visualize clear-cut stages of implantar colonization of a high virulent Sitaminum isolate SS97009 by tagging it with GFP using P-NIST plasmid. The methodology is as follows: isolation of distinct mating types and then followed by protoplast isolation from both MAT1 and MAT2. Then GFP tagging by polyethylene protoplast transformation is done, followed by confirmation of GFP tagging, microscopy and PCR. Further, testing of the mating ability of the transformants in vitro and visualization of implantar colonization stages in sugarcane was also done. Coming to the isolation of distinctive mating types, surface sterilized teleospores were surface sterilized and plated onto PDA with streptomycin to obtain mycelial colonies, and a single mycelial colony was again plated onto PDA agar again to obtain haploid colonies. The opposite mating types were identified by random mating method, which is based on a fuzzy type appearance on the plate. And further, confirmation of distinct mating types was done by PCR targeting B mating type genes. After that, protoplasts were isolated from the distinct haploid sporidia by treating with the lysing enzymes from Trichoderma hasianum and beta-glucanase enzymes. So this resulted in clearly visible and viable protoplasts, and the viability of the protoplasts were confirmed by using propedium iodide stain. So before proceeding with the PEG-mediated transformation, we have checked the regeneration of the protoplast also. And the protoplasts isolated, the viable protoplasts were again incubated with the P-NIST plasmid, which is harboring an EGFP and hygromycin gene, and incubated at 28 degrees Celsius for 5 to 7 days. The transformants grown on selection media were analyzed under epifluorescence microscope. So here we have got bright fluorescence with the transformants, but no green fluorescence was there with the wild type isolate.
Further, the expression of GFP was done during distinct mating types developmental stages in vitro was confirmed. So the GFP tagged haploid sporidia were able to fuse and form dichoritic mycelia on plate. And these were the timings of the key events in the distinct developmental stages in vitro were analyzed, and conjugation hyphae was observed after 4 hours, followed by haploid fusion at 8 hours, and initiation of DM was started after 12 hours, and mycelial clumps were observed after 15 hours. And further, the GFP tagged haploid sporidia were confirmed by PCR using gene-specific primers, GFP and hygromycin. Finally, the visualization of GFP during implantar colonization stages were done after inoculating the pre-germinated buds of a susceptible variety CO97009 by dip inoculation and syringe inoculation method. So here we have with the GFP tagged haploid sporidia, we got a BIP indicating its pathogenicity. So during the initial stages, that is 2 to 10 dpi, extensive colonization of the bud surface with the haploid sporidia and DM was seen. And also we got we were able to see intracellular colonization, establishing the penetration of the mycelia into the sugarcane tissues. But there was no fluorescence was observed with the wild type. And during 8 dpa, that is during BIP emergence stages, we can see the sporogenesis in the peripheral portions of the meristem, and in the central part of the meristem, colonization of parenchyma cells as well as vascular bundles could be seen. Coming to the summary, the study presents an efficient transformation method for acetaminium GFP tagging. The timings of the key events of life cycle stages of acetaminium in vitro were determined using the GFP tagged haploid sporidia. The GFP expressing isolate enabled direct and precise detection of implantar colonization stages of acetaminium in sugarcane. The development of transformants by protoplast-based PEG-mediated transformation will also serve as a tool for functional characterization of genes through homologous recombination to decipher molecular mechanisms of plant pathogen interaction. Authors greatly acknowledge Department of Biotechnology for funding and Director, Sugarcane Breeding Institute for facilities. Thank you.
Okay, Agisha. Agisha, you have taken too much time and also more slides presented. Slide is a number of slides restricted to 5, but
Yes,
work is somewhat elaborated, and
yes,
still is a putative or confirmed transgenic isolates it have developed.
yes, sir.
Hello.
Yes, hello, sir.
You have any promising transgenic plants after transformation? Hello.
Hello, sir.
You have protoplast fusion.
Yes, sir.
And you did the transformation.
Yes, sir.
So you have any promising isolates you have?
We didn't. We have checked the pathogenicity of the isolates, sir. And we have used it for visualizing implantar colonization stages in sugarcane.
Okay. You have haploid.
Both the haploids. Yes, sir.
Okay.
And the one which is having brightest fluorescence, green fluorescence, was selected for this implantar colonization studies.
Yeah, it's a confirmation. It's a
Yes, sir.
illuminate the confirmation of a transgenics.
Yes, sir. Okay. We have also confirmed by PCR using gene-specific primers, both GFP as well as hygromycin.
Okay. Thank you, Mitch.
Thank you.
Next presenter, Amala Mol from Sugarcane Breeding Institute. Can you please present?
Yes, ma'am.
Yeah.
Sir, can I start?
Yes, proceed.
Good evening, one and all. Myself, Amala Moldi from Sugarcane Breeding Institute, is here to present my research finding regarding transient ectopic expression of two candidate genes, CFEPL1 and CFPDIP1, in the model system tobacco. EPL1 is eliciting plant response like protein 1, and PDAP1 is plant defense inducing protein 1, which are the pathogen signatures of C. falcatum, the causative of Colletotrichum falcatum, the causative of red rot, which is a major menace to sugarcane cultivation in many countries in the tropics span the globe. In the present scenario, wherein which many elite varieties often succumb to the red rot pathogen, as well as the application of fungicides raises ecological concern, there is always a pertinent need to come out with the novel strategies like pathogen-derived resistance, induced resistance strategies, which can go hand in hand with the existing strategies to engineer durable red rot resistance in sugarcane. So the success of pathogen-derived resistance strategies relies on identification and proper characterization of pathogen-derived molecules which have the potential to induce resistance in the host. In the present study, we have attempted to functionally characterize two candidate genes, CFEPL1 and CFPDAP1, with a special emphasis on their in-plant localization as well as the defense inducibility in the model plant tobacco. To begin with, the coding sequences, protein coding sequences of protein coding regions of EPL1 and PDAP1 together with the signal peptides were amplified from the cDNA of a well-sporulated culture of CFEPL1, which is the virulent isolate, and the amplicons were cloned into the MCS of Picambia 1302, which is a plant-based. Excuse me, ma'am. Pardon. The amplicons were cloned into the MCS of Picambia 1302, which is a plant-based binary vector, and the recombinant vectors Picambia 1302 EPL1 and Picambia 1302 PDAP1, together with the control, that is the empty vector control Picambia 1302, was electroporated into the agrobacterium strain LBA4404 and was introduced into the tobacco via agroinfiltration. Samples were collected at different time intervals for microscopic observation for monitoring the co-localization of gene GFP fusion constructs, also visual observation for hypersensitive responses, as well as arrows accumulation was also monitored, which was followed by comparative transcriptome analysis for defense-related genes in the model host tobacco. Moving on to the results. Agroinfiltrated samples collected 60 hours post-infiltration when used and when viewed under fluorescent microscope in a GFP filter, we could see that the samples expressing GFP alone, that is the control, the fluorescence mainly localized to the nuclei as well as the extracellular surfaces, whereas in case of the gene GFP fusion construct, they got localized exclusively only to the apoplastic or the extracellular surfaces alone. This goes in line with the predicted apoplastic nature of these two proteins using apoplast P, which is an in silico tool. And the visual observation for hypersensitive response was done 5 days post-infiltration. And we could see that PDAP1 expressing samples scored maximum hypersensitive response 5 days post-infiltration when compared with the EPL1 expressing and GFP alone expressing plant samples. Again, DAB staining was done to monitor the ROS accumulation, the reactive oxygen species accumulation. We can see that prominent brown precipitate was formed in the gene expressing, that is EPL1 and PDAP1 expressing samples. Brown precipitate formed due to the oxidation of DAB by the reactive oxygen species catalyzed by the peroxidases. Following which we went for the transcription profiling of the various defense-related genes in the agroinfiltrated samples collected, monitored during different time intervals. Out of 13 genes studied, we could see that 9 genes showed remarkable upregulation during different time intervals. In this, the comparative transcriptome analysis was done using delta-delta CT method, and the experimental control used was the Picambia infiltrated, that is the empty vector infiltrated leaves, and the internal control was NTL23, ribosomal protein coding gene. So here we could see that the PR1, PR5, and PR4 AVR, which are pathogenesis-related genes, got upregulated. Then PAL1 and CHN50 both are salicylic acid pathway-dependent genes, which also got upregulated when compared with the control samples. PAD4 and EDS1 both are effector triggered immunity signaling pathway related genes. They also got upregulated. as well as defense-related transcription factors, WRKY and MIB protein coding genes also upregulated in the EPL1 and PDAP1 expressing samples. Moving on to the summary, the implantal localization of EPL1 and PDAP1 further reaffirmed and substantiated their predicted apoplastic nature using apoplast P in silico tool. And when transiently expressed, these two proteins could induce hypersensitive responses as well as ROS accumulation, and also upregulated defense-related genes in the model host tobacco. This experiment is actually a prelude, is an introduction towards stable expression of the candidate genes in sugarcane. Thank you. I thank the society for giving me this opportunity. Thank you everyone for listening.
Is a pathogen-related protein, PR125. Is it oscillated in a stable form or in a different?
Yeah, so they oscillated. Actually, it was the maximum upregulation we could see in 96 hours post-infiltration. And the gene PDAP1 showed an overhand over EPL1 expressing and the GFP alone expressing samples. In case of these genes, pathogenesis-related genes.
How many pathogen-related genes you have detected?
I'm now working on 2 pathogen-related genes, EPL1 and PDAP1. In the previous study done by our lab group, we have identified almost 11 genes, proteins, which are differentially expressed in pathogenesis-related, pathogen signatures. Which are
13 genes identified, out of which 9 is promising. 9 genes already expressed in the presentation.
No, why? Excuse me, sir. Yes.
Yeah.
Out of 13 genes, 9 differentially. There was some marking difference between the expression. Rest were like I could not see much upregulation.
Okay. See you. Thank you.
Thank you, sir.
So, if no questions.
Next, Amala.
Yeah, we can. Ashwati from Rubber Research Institute of India. Please start your presentation. Yeah, Ms. Ashwati, are you here?
Yes.
Yeah, please start.
Sir, can I start?
Yes.
Okay, good afternoon all. Myself Ashwati, first year PhD scholar in Rubber Research Institute. Today I am going to present my topic towards unraveling the interplay between salicylic acid and jasmonic acid pathway in Hevea phytophthora interaction. Hevea brasiliensis is one of the major crops which produce viable quantities of rubber. Hevea is infected by many pathogens, among which abnormal leaf fall caused by phytophthora is one of the important diseases which cause major yield loss. We all know that during host pathogen interaction, to counteract the effects of pathogen activity, plant activate many defense mechanisms like mechanical barriers, anatomical barriers. When these first line of defense get fail, the plant activates second line of defense, which mainly include salicylic signaling pathways such as salicylic acid pathways and jasmonic acid pathway, mainly during biotic invasion. So I briefly explain the signaling both signaling pathways, salicylic acid pathway and jasmonic acid pathway. Please have a look into the PPT. During biotic invasion, salicylic acid get upregulated in the plants. It will combine with the NPR1. It will combine with the NPR3 and 4, and during that time the NPR1 get released from NPR3 and 4. Then NPR1 is a key regulator for salicylic acid responsive genes. The free NPR1 will combine with other transcription factors, and it causes the synthesis of salicylic acid responsive genes like PR1. So the key regulator of salicylic acid pathway is NPR1. In the case of jasmonic acid pathway, during necrotropic invention, jasmonic acid gets synthesized and it combined with the coronadine insensitive 1. The action of coronadine insensitive 1 is to degrade the JAS responsive gene repressor called JAS. The function of coronadine insensitive to degrade the JAS repressor. After the degradation of the JAS repressor, jasmonic responsive genes get activated. So in Hevia already reported the effect of salicylic acid by the exogenous application of salicylic acid, and it is found that PR1 and other genes are activated, but no other studies where no other information is related to the role of jasmonic acid in Hevia. So in our study, the primary aim is to find whether this salicylic acid pathway and jasmonic acid pathway is acting synergistically or antagonistically. For this, this is in our study, this is found out through an transcriptomic approach. For this, we prepared the transcriptome of a resistant and susceptible. We prepared the transcriptome of resistant and susceptible clone of rubber in control and the disease conditions, and using bioinformatic approaches, first we identified all the NPR gene, that is the key regulator of salicylic acid pathway. First, we identified the all the NPR genes involved in salicylic acid pathway, and it is found that two NPR genes showing highly upregulation in both diseased conditions, and their corresponding fold change values are provided in the slide, and it is identified that these NPR genes are mainly involved in salicylic acid pathway. Next, in order to find out jasmonic acid pathway is acting synergistically or antagonistically with the salicylic acid pathway, we search for the regulation of coronadine insensitive gene, that is the key regulator of jasmonic acid pathway, and it is found that in both clones after treated condition, coronadine insensitive gene is down regulated, which shows that salicylic acid pathway has some role in suppressing jasmonic acid pathway. And so the relevance of this study, future quantitative validation using real-time PCR is in progress to conclude our result. The relevance of this study is if salicylic acid pathway is suppressing the jasmonic acid pathway, there is a chance of there made a chance to the invasion of other necrotropic pathogen to attack the plant, since it is the jasmonic acid pathway is suppressed by salicylic acid pathway. So we have to find how this jasmonic acid pathway is inhibited, how this coronadine insensitive gene is suppressed during the activation of salicylic acid pathway. These findings will help us to identify how the resistant mechanism in plant is acting. So unraveling the interaction between both pathway help to understand the difference in resistance in both clones, and if we can rewire the salicylic acid pathway and connecting jasmonic acid pathway, we can identify more information about both pathways and its role. Thank you.
In your presentation, is a salicylic acid pathway is a suppressing jasmonic acid pathway, is vice versa. So what kind of PR proteins is developed?
Yes.
Pathogen related
Proteins.
You have identified PR protein. You have mentioned the pathogen related protein identified. What kind of pathogen related protein?
No, in the transcriptome data, we got many, we identified many pathogen related proteins. So we conclude that pathogen related proteins are upregulated. For this study, we took only the the genes that is involved in the salicylic acid pathway. No, further work are in progress. Okay.
So you have presented so many slides. Actually you have to restrict the number of slides only 5. But you have taken more time. Okay. See you next.
So next presenter Chandrika Ghoshal. Are you there?
Yes, ma'am.
Yeah, please.
Can I start? Am I audible?
Yeah, yeah, you can start. Chandrika, it's audible.
Okay. So, hello all. This is Chandrika Ghoshal from Division of Vegetable Science, IRI Pusa. My topic is on identification of anthracnose disease-resistant chili lines in India. Chili is an important vegetable spice crop grown in India. Chilies are rich in vitamin C and vitamin A content. The active component of chili, capsaicin, has antioxidant, anti-carcinogenic, and anti-mutagenic properties. India is the third largest producer of chili in the world, but around 54% production loss is caused due to anthracnose disease. Anthracnose disease is mainly caused by the fungus Colletotrichum. It causes yield reduction and quality loss of chili. Symptoms appear as shrunken, circular or angular lesion on ripe chili fruits. Brown spots with black margin can also be seen on the leaves and the stems. To combat this problem, large number of fungicides are in use, but these causes pollution and health hazard as well. Therefore, it has become necessary to identify resistant lines of chili anthracnose that can be used in breeding program to develop resistant variety in India. So the objective of my research work is to identify anthracnose disease resistant lines in chili. So these are the three Colletotrichum cultures which I have used in my study. These are Colletotrichum gloeoporoides, Colletotrichum brevisporum, and Colletotrichum capsaicin. So in this experiment is done in laboratory as well as field condition. A total 169 chili lines are used in this experiment. For artificial screening in laboratory, the healthy green or ripened fruits are first detached, and then the inoculum was prepared at the concentration of 5 into 10 to the power 5 conidia per ml. Then the inoculation is done at two sites using microinjection method and incubated in plastic moisture boxes at high relative humidity and 28 degrees Celsius for 15 days. The lesion diameter and the fruit size were measured. The percentage of disease severity was calculated by dividing the lesion size to the fruit area. For artificial screening in the field, the same method of inoculation and scoring was done. The canopy of the inoculated plants were watered from next morning up to the 1 week to stimulate conidial differentiation. Fruits 169 lines, and all the 169 lines were infected with three Colletotrichum cultures, that is Colletotrichum capsaicin, Colletotrichum brevisporum, and Colletotrichum gloeoporoides. All the lines were susceptible against Colletotrichum capsaicin and Colletotrichum brevisporum, but with Colletotrichum gloeoporoides, I have got 3 resistant and 3 moderately resistant lines. This is a picture of 2 chili lines, one susceptible and one resistant against Colletotrichum gloeoporoides. This picture is taken 15 days post-infection. When infected with Colletotrichum capsaicin and Colletotrichum brevisporum, fruits of both the lines got completely infected, but with Colletotrichum gloeoporoides, DC 146 is showing resistance. So the conclusion of my studies are all the lines are screened against Colletotrichum capsaicin and Colletotrichum brevisporum show highly susceptible reaction. 3 lines were moderately resistant and another 3 lines show fully resistant reaction against Colletotrichum gloeoporoides. So we can conclude the virulence of Colletotrichum capsaicin and Colletotrichum brevisporum were more than Colletotrichum gloeoporoides. So the significance of this study is the resistant lines can be used in breeding program to develop resistant varieties in future, which might be more economic and eco-friendly. Thank you.
In your presentation, you have shown that Colitotrichum brevis strain
Yes, sir.
D146 highly resistant.
Yes.
But in conclusion, you have given only moderately resistant. So is
No,
correct.
sir. In 3 lines are moderately resistant and 3 lines are completely resistant.
But in conclusion, you have not shown that completely resistant.
No, sir. In the conclusion also, I have shown that 3 lines are. In the picture.
Yeah, moderately resistant.
Yeah. In the picture, I have given only the susceptible line and one picture of the resistant line.
You shown that D146 is a highly resistant.
Yes, sir.
Thank you. Next. Any discussion? Any questions? Thank you very much. Yes, next one, Gopi. Gopi.
Yes, sir. I'm here, sir.
Okay.
Good evening, all. Now I am going to present the posted topic status of diseases in sugarcane germplasm at Kannur. Sugarcane is one of the most important crops belonging to the family Poaceae, cultivated both in tropic and subtropic regions of the country. ICS Sugarcane Breeding Institute Research Center Kannur, established in 1961, maintains largest collection, about 3,373 germplasms, belonging to 6 saccharum species and different hybrids, IA clones, and allied genera. Diseases are the major concern in sugarcane. More than 55 diseases have been reported in sugarcane. Smut, wilt, pocoboing, set rot, sugarcane bacilliform virus, rust, ring spot, stalk rot, OLD are the common diseases reported from SBRC Kannur. So coming to the materials methods, it's a simple study only. So regular monitoring at monthly interval was done. Number of infected.
Your slides are not coming.
Doctor, slides are not visible.
Not opening.
Yeah, not yet opened.
Okay, sir.
Please redo it, restart. First you open your slides.
So I opened, sir.
Here it's not coming. Share it. Here only your pen drive files are showing. Now, if you have any problem, you can do it and we can start with the next presenter.
Okay, sir. Okay, sir.
Madam.
Next presenter is Mr. Javeed from GKVK Bangalore. So, Mr. Javeed, are you available?
Yes.
Yeah. If you are ready, you can start.
Can I present now?
Who is presenting?
Javed. I cannot share.
Yeah, Javed.
Hello.
Dr. Gopi, you unshare your screen.
Okay, sir.
Mr. Gopi, you can close your screen. Hello?
So there is some problem, sir.
Okay.
Okay. Is it visible now?
Yes, visible.
Okay. Good afternoon to all. Myself, Javed Habibi. Mainly, I'm studying in plant pathology, College of Horticulture in Bangalore. My topic research is screening of cucumber varieties, hybrid for rotten method, Maladigina incognita, and management. Introduction. Cucumber is the most important crop, vegetable crop in the world after tomato in full crop, which have a lot of diseases. Among all diseases, plant parasitic nematodes is also important. Among the plant parasitic nematodes, Rotna nematode is the most important disease which distributed worldwide, especially Maladigina incognita, which among around 80 to 90% of all damages caused by Maladiguna species in tropical and subtropical regions. The objective which I conducted the screening of cucumber variety for Rotna nematode and management of Rotna nematode under Paleos condition. The material and method which I used first initially after perennial pattern of the nematode, which I identified the Maladigina incognita species, I multiplied them from single egg mass in tomato. Then 19 variety of cucumber I screened after extracting of nematode and released of them to the 90 variety. After 15 days of the seedling transplanting of the seedling, I released 2,500 second germinals of Rotna nematode in the soil surrounding of the seedlings. Then the severity of the disease was recorded after 90 days of sowing. The treatments for management which I used, 3 bioagent, Trichoderma arzonium, Petunia colomadisperia, Pseudomonas florensis, at the rate of 10 gram per pot, and also 1 nematicide by name of Valium prime or flupirum at the rate of 0.3 ml per 1 liter of water. And also the number of 1,500 second germinal of Maladiguna species are released to manage by 3 bioagent and nematicide flupirum. The result which from my screening which I got during the growing of the plants symptom was extended plants, patchy growth and yellowing of leaves, heavy root galling, poor yield, weak plant, small slow growth fruit. And also during this my screening, all of this 19 varieties and hybrid were highly susceptible, and none of them found highly resistance or immuno. The most highly susceptible then I selected by name of Pahlavi hybrid and for my management studies. From the management studies, my observation was the plant growth parameter and nematode parameter. And from this result, it's revealed that among all these treatments, combination of nematicide and also this bioagent showed the best result compared to the individual of them. Here is showing treatment 11 and also treatment 12 for plant growth parameter. They showed best result compared to the individual of bioagents or nematicide. Also effect of this treatment, different treatments on nematode multiplication. Among also this also showed the combination of bioagent Trichoderma arzonium, Petunia colomadisperia, and also flupirum showed best result for control of Maladigina incognita in cucumber. Conclusion. Based on this research, I concluded that combining of bioagent and chemical for management of Malaria incognito was enhanced the plant growth parameter and significantly reduced the nematode population and disease severity under pot condition in foliars. Also, it's an effective environment-friendly approach to an important compared to component to manage Malaria incognito. Thank you all.
So in your conclusion, you have concluded that is an environment friendly approach. What you have included is a nematicide.
Yes, sir.
Is a very good 4 combination or already proved that you are very good in controlling the nematode. But too many combinations is very difficult to adapt by the farmers.
Yes, sir.
Is the 4 combination is not able to adapt the by farmers, is a very difficult to disseminate the particular technology to the farmers.
Yes, sir.
So we have to restrict the number of combinations whenever go for recommendations.
Okay,
so for eco-friendly management, what are the other options we have to explore? We have included 4 different treatments for the conclusion is successful in controlling the nematode. So we conclude that number of treatments have to reduce.
Okay, sir.
Too many combinations should be avoided.
Okay. But based on my results, sir, I found the combination of this nematicide with bioagent that show best result. And also trichoderma arsenium with that flupirum or valimpuron, they showed best result to control the nematode.
Maybe result may be good, but is adaptability is a big question mark.
You are right, sir. You're right. Thank you.
Thank you. Next.
Javid. Hello, Javid.
Yes, sir.
I want one clarification.
Yes, sir.
You combined the 3 different things, one bacteria, one fungi, and one nematicide. Have you tested its compatibility before application of this into soil? Its compatibility under in vitro?
No, sir, I did not test them. Directly, I mixed them before transplanting of the cucumber and soil. But I did not test.
Okay. Before going for a field application or any in vivo studies, this has to be tested for their compatibility under laboratory conditions, so that their effectiveness.
Okay,
or as Dr. Devanathan told, too many combinations may be avoided. Okay, thank you.
Okay, sir. Thank you.
Sir, shall I do, sir? Gopi?
Gopi, sir, start your presentation.
So myself, Gopi, shall I start, sir?
Okay. Proceed.
Sir, slides are visible, sir?
Yes.
Okay. So good evening again to all of you. Now I am going to present my poster topic, status of diseases in sugarcane germplasm at Kannur. Sugarcane is one of the most important crops belonging to the family Poaceae, cultivated in both tropics and subtropics of the world. ICR Sugarcane Breeding Institute Research Center Kannur, established in 1961, maintains largest collection of germplasm. More than 3,373 are maintained here. It consists of 5 Saccharum species and different hybrids, IA clones, and allied genera. Diseases are the major concern in sugarcane. More than 55 diseases have been reported in sugarcane. Most importantly, like smut, wilt, pocoboing, etc., sugarcane bacilliform virus, rust, ring spot, and stalk rot (OLD) are being reported from ICR Sugarcane Breeding Institute Research Center, Kannur. Coming to the materials and methods, here only germplasms are monitored regularly in monthly interval, and a number of infected clones and disease incidence and disease severity was studied, and further management measures have been done. So coming to the first disease, this is the most important disease in sugarcane germplasm at Kannur because of high relative humidity and rainfall. So it has been observed in almost all the clones, so to the extent of 100%. But however, only in Saccharum robustum, the incidence, percent disease incidence was comparatively less when compared to other clones. So some clones in Saccharum barberi and Saccharum robustum in some years free from this disease. 4 Saccharum officinarum clones, the ring spot was more severe, like Mongetgam, NOS IK767 and white transparent. It reported more than 75%. So these are the symptoms of the ring spot disease. And sexual stages Leptospirea saccari and other asexual stages Philosticta saccari. But one interesting thing is that, so among the clones, it was found more in case of leaves, clones with a dark color leaves.
Go ahead, Kobe.
கீழே என்னோடதுலயும் கொஞ்சம் மைக்கை வந்துட்டு செட் பண்ணணும். என்னோட கேமராவும் இதுவும் வந்து சரியா வர மாட்டேங்குது. அதனாலதான் இங்க ஓடி வந்து உட்காந்துருக்கீங்க.
Hello. Proceed, Kobe.
Gopi, please unmute your mic. So, Mr. Gopi, please unmute your mic.
Set it, sir. I'm audible, sir.
Yes.
Set treatment with hot water and carbendazim and prolonged flooding with the plant debris might be the reason for the absence of the disease in the beginning, because normally disease appear at the end of the season. So another important is the Pocoboang. It is caused by the Fusarium saccari. Only 3 groups like Saccharum officinarum, foreign hybrids and Indian hybrids were found infected. Among them, Saccharum officinarum and foreign hybrids contributed more. Like 2019, it was in Saccharum officinarum, 8 clones were infected. In case of foreign hybrids, 7 clones 2017 were found infected. So here all the 3 phases like chlorosis, top rot and knife cut symptoms were observed. Top rot was observed in the case of IG7670 and a knife cut symptom was observed in the case of 4002. So another important is the Puccinia melanocephala. So it was found in the case of 4 groups: Saccharum officinarum, foreign hybrids, Indian hybrids, and Saccharum spontaneum. Among the clones, Indian hybrids experienced more. We can find that like 2017 only it was 3, but it has increased to 32 clones. So that indicates that it has more infected clones when compared to the other clones. So similarly, Saccharum spontaneum clones belonging to the IND clones, that is 81, 20, 74, 82, 83. In all the seasons, we have found this disease. In case of Indian hybrids, Co376 experienced more disease severity, and also followed by Co698 and 699. So another important disease is a stock rot. It was found first in our station only by Viswanathan and his co-workers. 3 groups: Saccharum officinarum, foreign hybrids, and Indian hybrids were found infected with this particular disease. However, Saccharum officinarum contributed more. We can find in 2017 it was 10, in 2018 36, and 2019 15. The percent incidence ranged between the 15% in case of Goyam A and 100% in the case of 51NG126. Viral diseases, various viral diseases like sugarcane bacilliform virus and sugarcane yellow leaf disease were found in germplasm, like Castilla and Listada, and also D1135 and D1135 STR are more common. Bacilliform, they are more common for the bacilliform virus. So other clones like 4 clones of Saccharum officinarum like NG10, NG7731, NG7761 and IJ76552 were found infected with the OLD disease. Similarly, 2 hybrid clones and 3 Saccharum robustum clones are also found infected. Maximum percent OLD incidence was found in case of MOL 4503. Coming to the conclusion, it is very clear from the study that major diseases like red rot and mosaic are absent. Other diseases are also not severe in germ plasm, which make SPRC Kannur as an ideal place for conservation of sugarcane germ plasm. This may be also due to the strict quarantine measures we follow and absence of sugarcane cultivation nearby, proper monitoring and disease management. However, in recent years, some new diseases like banded sclerosis disease, sclerotial rot, red stripe, and flower smut are noticed in few clones, maybe due to change in weather parameters, mainly rainfall pattern and frequent floods during the 2018 and 19. Here also we can see the rainfall is comparatively less in 2018 and 2019 when compared to the 2017. We also experienced floods during 2018 and 19. So this is a flooded condition in our station. So this is the disease, banded sclerosis. This is first time we have reported from our center. Thank you, sir.
So is any weather factor influencing the susceptibility of cocoa bomb?
Sir, actually, we could not correlate the. I have analyzed the data, sir, but I could not find any conclusion regarding the weather factor, sir. Correlation was actually like less.
Epidemiology is the most important.
Yes, sir. Yes, sir.
Outbreak of a particular disease.
Yes, sir. I could not correlate. And other thing is that like the clones which are found infected during 2017, I could not found again infected by the same disease, sir, especially in case of Pocoboy.
Okay. So open for discussion. Okay, so next one, Jayalakshmi. Jayalakshmi, please. So close your PowerPoint, sir.
Sven, நீங்க கொடுக்கல Sven.
Chairman, sir, I think Jayalakshmi is not there. Please.
Lakshmi is still getting late. Not I will possibly can proceed to next one.
Ms. Limia Joseph, sir, from Rubber Research Institute of India, Kerala. I think she is ready. So, Ms. Limia Joseph, are you ready?
Yes, ma'am. Yes, ma'am. Can you hear me?
Yeah, you are audible.
A warm good afternoon. Can you hear me?
Yes,
Yeah.
yes, can you?
Good afternoon to all who are listening me. My topic is linkage mapping and genome-wide association studies as potential tools to discover regions of disease resistance in rubber genome. Hevia brasiliensis, as we all know, the para rubber tree is an economically important crop as it produces latex of commercial utility. Several diseases or pathogens cause significant reduction in its growth and latex production, leading to significant economic loss. Phytophthora, Corinospora, and Colletotrichum are the three major pathogens reported in rubber tree. Even though various chemical and biological control measures are available to manage this disease, cultivation of disease-resistant varieties is a promising eco-friendly alternative. Marker-resisted selection is the principal and the preliminary step behind resistance breeding. Use of DNA markers that are tightly linked to disease-resistant loci, that is marker-resisted selection. For marker-resisted selection, identification of markers or QTLs linked to the trait of interest is necessary. That can be achieved by two approaches, namely linkage mapping and association mapping. However, both these methods result in the identification of QTLs related to a trait of interest. So, an integrated application of this method, a combined linkage association mapping method, can be used to detect all the QTLs linked with a trait. First is linkage mapping. For linkage mapping, we developed a mapping population by an interspecific crossbreed between Hevia brasiliensis and Hevia benthamina. As you can see, these parent plants differ contrast in their basic characteristics like yield and disease resistance. After developing a progeny population, both the progeny population and the parents were genotyped and phenotyped. Genotyping was conducted by a DAT sequencing method. It's a genotyping by sequencing method. And phenotyping at the same time conducted separately for the pathogen Phytophthora, Corinospora, and Colletotrichum. For Phytophthora, phenotyping done by leaf disc assay, and for Corinospora and Colletotrichum, toxin-based assay was followed. From the genotypic data derived from the DAT sequencing, we were able to develop the linkage map of both the parents, indicating, and it consists of 18 linkage groups. Finally, by combining the genotypic and phenotypic information or merging this data into a software called QTL, we identified the major QTLs related to disease resistance in Hevia. In the case of Phytophthora, we identified two QTLs respectively on linkage group 1 and 8. For Corinospora on linkage group 1 and for Colletotrichum on linkage group 18. The exact position of the QTL identified and their lot score is given here in the linkage map. Then to proceed further or to study more about these QTLs, to study broad about the transmission of a marker or a QTL at the population level, we started or initiated genome-wide association studies or association mapping. For association mapping, we are using a set of clones, namely VCAM clones, which are diverse, unrelated cultivars. As we see in the last slide, for association mapping, association mapping can be followed in a natural or raw, unrelated population. However, for linkage mapping, we have to develop a mapping population prior to genotyping or prior to mapping. So for association mapping, we selected VCAM clones. These VCAM clones were evaluated in the, these VCAM clones were evaluated in both in vitro and in vivo. screening was conducted and the phenotypic extremes for each of the pathogen was selected and the association panels were developed. That is, association panel for a particular pathogen consists of the highly resistant clones and the highly susceptible clones. These, the association panels were they later subjected to DNA extraction, and the pooled DNA was subjected for genotyping and population structure analysis. Currently, we are in the analysis part of association mapping. From this work, from both linkage mapping and association mapping, we hope to identify all the QTLs linked with the disease resistance in Hevia, so that we can use those markers or QTLs for molecular screening of rubber clones prior to their release for cultivation. That's all about my topic. Thank you, sir.
Is open for discussion. Any marker as the resistant genes I identified?
Yes, excuse me, sir.
These two clones. One is highly susceptible. Another one is moderately susceptible.
Okay.
Is a tolerant. Is there any resistant line or still tolerant?
There are not, we can say it's completely resistant. We are using, it's more common to say, or it's more useful to say tolerant one. There is no clone 100% tolerant or highly 100% susceptible. That's why we are using a group of clones for association mapping.
But is a QTL parameters, how it looks? Is it promising or high yielder?
We are using this QTL as a marker. If the QTL or the marker specific, if the marker is there, the plant will be resistant. Yeah. We are proceeding in such a way, and we identified the QTL for each of the pathogens.
Okay. Thank you.
Okay, thank you.
What about the next open for discussion? Okay, Jayalakshmi, next.
Good evening all. Myself Jayalakshmi. I am from Sugarcane Breeding Institute, Coimbatore. Here I am with a poster presentation. The topic is development of an efficient method for apoplastic protein extraction from Sporosaurium citaminium infected sugarcane meristems. So you can know that sugarcane is industrially important for sugar production. So around 80%. Around 80% of the world sugar production is from India. But nowadays, the sugar production decreases due to many of the diseases. Smut disease is one of the major diseases that is caused by Sporosaurium citaminium. The characteristic symptoms of the sugarcane smut disease is the development of whip-like structures in the sugarcane plant. So we can know that the Sporosaurium citaminium, since it is a biotrophy fungus, it have a long-term relationship with this apoplastic region of the plant. So this Sporosaurium citaminium colonized in the apoplastic region of the plant and secreted its effectors into the plant apoplastic region. So in that apoplastic region, plant in turn secretes its resistant gene into the apoplastic region. So this apoplastic region is the real war zone between these two biomolecular barriers. So in order to study the apoplastic proteins, we have to first isolate the apoplastic proteins to study this plant pathogen interactions. So that as of now, there is no standardized method for the apoplastic protein extraction. So in our study, we are aim to develop a method for the extraction of apoplastic proteins. So for that, we first collect the meristem tissue from the infected sugarcane plant, then subjected into syringe infiltration and vacuum infiltration. We are using a sodium phosphate buffer, potassium phosphate buffer, calcium chloride plus sodium acetate buffer, and distilled water for this infiltration process. And after the syringe infiltration and vacuum infiltration, we collect the wash fluid and subjected into quantitative analysis as well as qualitative analysis. In the quantitative analysis done by Bradford method, while checking the result, we can see that around below 30% of the apoplastic wash fluid was collected, and the protein concentration in this fluid was in syringe infiltrate, and when we compare the syringe infiltrate and vacuum infiltrate, the protein concentration is almost same concentration. And we done the qualitative assessment of the extracted proteins. After acetone precipitation, we done that SDS page. From this profile, we can say that we have to move for the cytoplasmic contamination, because we focus on the apoplastic proteins. So we have to minimize the cytoplasmic contamination in our extract. So for that, how much is our cytoplasmic contamination? So in order to know that, we move for the qualitative analysis with biochemical assays of this with cytoplasmic markers. For this cytoplasmic analysis, we took malate dehydrogenase assay and glucose 6-phosphate dehydrogenase assay. So in these two assays, we can see that apart from sodium phosphate, except sodium phosphate, other buffers give more less cytoplasmic contaminations. So we have to focus less cytoplasmic contamination in our apoplastic proteins. So the buffers and methods which prefer less cytoplasmic contamination, which give less cytoplasmic contamination, we prefer that method. So we can conclude that in vacuum infiltration and syringe infiltration, the syringe infiltration method is an effective method for the apoplastic protein extraction. It gives less contamination, less cytoplasmic contamination. So in order to standardize the buffer, to selecting an appropriate buffer, the quality of extraction is being further evaluated by a more stringent method. Thank you.
So in your presentation, you have standardized techniques for the extraction, for plastic extraction.
Yes, yes.
Is prove that the syringe method is highly suitable for the easy extraction of plastic.
Yes, yes. Syringe method is give less cytoplasmic contamination.
Less cytoplasmic contamination.
Yes, yes.
Okay. So we have to, yeah. So then what will the next level of approach, future plan?
So we have to then we have to do the Western blotting, so that we can refer in check these three buffers. So we can know that which is the giving less cytoplasmic contamination. We can select that buffers for the extraction process. Then we can continue with the extraction and identify the proteins and
A particular reaction specific protein.
yes, sir.
Is a,
Yes, sir.
under Western blot.
Yes, sir.
Okay. Thank you. Next.
Thank you, sir.
Open for discussion. Sir, any questions?
So,
Thank you. Next.
Lishma from College of Horticulture, Kerala. Trishur. Lishma, are you available? Lishma from Villanikara. Sir, I don't think she's here, sir. Lishma? Okay. So can we move on to the next one, sir?
Priya Ravi Kumar.
Yeah, Priya Ravi. Priya Ravi Kumar from IRA Center, Nilgiris, Wellington. Priya?
Sir, Priya Ravikumar is not available.
Okay. And the next one is Rajeshwari from College of Horticulture, Bangalore, UHS, Bagalkot. She's available. Rajeshwari, she's available. So Rajeshwari, from Bagalkot. She's available, sir. So next one, Vikas from Wellington, IRA Regional Station, Wellington. Vikas. Vikas from IRA Wellington. Next participant, Vinodini from Department of Plant Pathology, TNI Coimbatore.
Yes, ma'am, I'm available, ma'am.
No, they start your presentation.
Yes, sir. Good evening, everyone. I am about to give my presentation on genetic structure and molecular variability of cucumber mosaic virus infecting chili in Tamil Nadu. So far, CMV is one of the most economically important virus infect most vegetable crop. This notorious virus causing severe mosaic disease in Chile. Hence, a diagnostic survey was conducted in different agroclimatic zones of Tamil Nadu to assess the prevalence of mosaic disease in Tamil Nadu. So the characteristic symptoms were observed as mosaic with a puckering symptom, mosaic with a leaf filiformity, mosaic mottling, and the stunted growth. So, based on the symptomology, T and CMV isolate have been these CMV isolates have been partially sequenced, and based on that, a representative isolate have been sap inoculated onto different host plants. So, to assess the symptomology of the TN isolates, so based on this, the symptomology, localized and the systemic infection have been observed in different host plants. The localized lesions were observed in the Nicotiana species, and the systemic infection observed in most of the host plants. And the presence of CMV in the inoculated plants have been confirmed through the RT-PCR and doc ELISA analysis. And then this is the genome organization of CMV, the complete genome sequencing, the RNA1, and actually the CMV is a tripartite genome, single-stranded RNA virus. So based on the sequencing, the RNA1 determined to be comprised of 3.3 kb, and RNA2 determined to be 3 kb, and RNA3 determined to be 2.2 kb. So RNA1 encodes a 1A protein, that is a replicase protein, which contains the 2 kb and translated into 93 amino acids. In the N-terminal region, it having the methyl transferase and determined till the 146 amino acids, and it having the C-terminal region with the helicase motif. Based on the sequence analysis, the more nucleotide variation were observed in the replicase protein in the central region from 1132 nucleotide to 2760 nucleotide, and also a specific amino acid substitution observed at the 461th position of replicase protein in the TN isolates, which is the determinant of the necrosis induction. So we correlated this amino acid substitution may have the correlation with the necrosis induction in the host strain study in Nicotiana plants, and followed by RNA2 encode 2A and 2B protein, which is a replicase protein, which contains it 2755 nucleotide. It having the conserved GDD motif for the synthesis of RNA-dependent RNA polymerase for the replication of the virus from 609 to 611 amino acids. And then 2B is a silencing suppressor protein, which determined to have the 336 nucleotide translated into 112 amino acids. There was a unique amino acid substitution observed at the arginine 46th position of 2B protein, which is attributed for the induction of mosaic and stunting symptom in the infected plants. And then movement protein, which contains the 840 nucleotide translated into 280 amino acids, which is vital for the systemic movement of virus. In the sequence analysis, we also observed the amino acid substitution from serine to arginine, but we didn't know the function of the amino acid substitution, and followed by the code protein, which is encoded from the RNA3, subgenomic RNA3, which contains 657 nucleotide translated into 219 amino acids, which is a fundamental determinant for encapsulation, symptom induction, aphid transmission, host range, and movement of virus. In this, we observed the conserved arginine-rich motif at the 5' end from 14th to 24th position, and also we observed the unique amino acid substitution from serine to proline in most of the TN-CMV isolates. Based on these amino acids, we can categorize the CMV into mosaic-inducing and chlorosis-inducing strain. And further comparative sequence analysis implicates that serine 129 and 309 at 137th position to be conserved in chlorosis-inducing strain, as like proline at 129th position, serine at 137th position to be conserved in mosaic-inducing strains of CMV. Similarly, lysine at 65th position and 82nd position conserved in among all the subgroup IB and subgroup 2 strains. Cumulatively, 30 amino acids observed to be conserved among the subgroup 1 and 2. Basically, CMV is categorized into subgroup 1 and subgroup 2. In subgroup 1, there is a category, it can be categorized into subgroup A and subgroup D. So there was a conserved deletion has been observed in the subgroup 1 strain at the 12th position. Similarly, conserved deletion observed in the 25th position of subgroup 2 strains. And then we did the pairwise identity matrix for TN isolates with the other subgroup IB, IA, and 2 strains. So from this, the subgroup, TN isolates showed the 97 to 99% of homology with the subgroup IB, and similarly 92 to 93% of homology with the subgroup IA, and similarly with 82 to 83% of homology with the subgroup 2. This is a graphical representation of pairwise identity matrix. And in the phylogenetic analysis, all the TN-CMV isolates, including chlorosis and mosaic-inducing strains, have been clustered into subgroup IB. So then the recombination analysis depicted that TN-CMV isolate may evolved from the black pepper isolate of Kerala as a major parent, and minor parent Egyptian isolate, tomato isolate of CMV. So overall, CMV is a notorious virus causing loss up to 45 to 60% in Tamil Nadu, and the maximum titer value of CMV observed as 1.5 to 1.7 in the dark ELISA analysis, and the complete genome of the virus has been characterized using the primers covering RNA1, RNA2, and RNA3 genome of the CMV. And from the nucleotide diversity analysis, we have observed that RNA2 is more variable than RNA1 and RNA3 segment of CMV, and code protein and movement protein observed to be less variable. And also the greater synonymous and non-synonymous substitution, nucleotide substitution was observed in the chlorosis-inducing strains of CMV rather than the mosaic-inducing strain. And further genome sequencing and the phylogenetic analysis revealed that mosaic diseases of Chile to be associated, to be induced, caused by the CMV subgroup IB with a mosaic-inducing and chlorosis-inducing strain. And also the evolution of these TN-CMV isolates were observed from the Indian black pepper isolate by the recombination between the position of 165 to 505 nucleotide. Hence, the understanding the genetic structure, diversity, and evolutionary history of virus important aspect for the development of management strategies. Hence, our study may give the better conception about the genetic structure and evolution of the CMV virus. Thank you.
So in your presentation, you have the identify the mechanism of mosaic-inducing strain. Is a method how it induces mosaic and chlorosis. Some strain inducing chlorosis, some strain inducing
Yes,
both
sir.
mosaic and chlorosis.
So no, sir.
So,
Amino acid, the serine at the 129th position of code protein inducing the chlorosis symptom. If it is
yeah,
substituted by the proline, then it will induce the mosaic symptom.
mosaic. Okay.
Yes,
Right.
sir.
So open for discussion. What's your next level of your approach?
Sir, this is a basic study, but our work is engineering the resistant against cucumber mosaic virus through RNA silencing technology. So preliminarily, we characterized the genome of the CMV. Now we are proceeding the RNA, developing the RNA construct to develop the resistant against CMV, sir. We have completed the complete genome characterization. Now we have developed the RNA construct, and we will introduce that into a plant and will evaluate the resistant mechanism, sir, through RNA.
So any questions? Open for discussion. Thank you, Vinodani.
So, thank you, sir. Thank you so much.
Next, Harita. Harita Jay Kumar. Meghana SP. Next, Soumya M. Agisha
Okay. ஒருத்தருங்க உள்ள போயிட்டாங்களா?
So session 1 also, I think out of 16, 10 they have presented, sir.
Hello, Tirupana.
Yeah, session
have
1,
presented?
madam. Yeah,
Okay.
out of 10 they have presented.
Okay.
So session 1 completed.
Okay. Everyone has presented. Is it?
No. 6 of them they are not there. Priya, Rajeshwari, Vikas, Harita, and Meghana, they are not available.
Okay. Thank you.
Okay, madam. Okay, thank you.
Hello, Raju.